So for today, we're going to be looking at the Bioconductor featured workspace. If you're following on slides, I provided a direct link to the workspace, but I'm actually going to navigate to it using some of the skills you learned today. So I'm going to go to app.terra.bio, and I encourage you to do the same. And I'll try to incorporate some pauses as we're doing this so that people can follow along, but definitely speak up if I'm moving too fast or if you encounter any problems that we can address as a group. Okay, once you're at app.terra.bio, you can do the same thing we did before. Go to your workspaces. And then go to the featured tab. So you see my workspace is new and interesting. Go to featured and search for Bioconductor. It actually pops up right away for mine. So I'm going to go ahead and open that. And then likely this workspace is going to be read-only for you. So we're going to need to make a clone of the workspace so that you can run the notebooks that are in it. So to make a clone, again, you're going to go to the three-dot icon in the right, to the right of the dashboard and select clone. And make a name for the workspace. I'm going to call it July 09 LK. And you would pick your billing project, workshop temp. And clone.
Sorry, Elizabeth, yeah. I missed where you selected the notebook or this workspace.
The workspace can, sure, let me just go back really fast. If you go to your hamburger menu, are you at app.terra.bio?
Yes.
Yep, so if you go to your hamburger menu and just go to your workspaces, you can go to the featured tab in your workspaces and then search for Bioconductor.
Oh, okay, thank you.
And then choose that one. Once everyone is here, we're gonna need to make the clone. So let's go ahead and do that. So as has been mentioned, it needs to be a unique name. Doing the date and your initials is helpful. And you should have workshop temp billing project. And then we don't need to worry about authorization domain because everything in this workspace is public access. So go ahead and click clone workspace. Okay. And let me just pause here for a second. The very first thing we're going to want to do is get our notebook runtime set up. Are there any questions? I heard some chatter. Okay. So,
a question, if we still have the previous notebook open, should we like close it or?
Yeah, I would go ahead and close out of that window. And we're going to start fresh. We're going to actually delete your runtime and everything. So if your runtime is still going, we're going to actually delete that virtual machine right now and create a new one.
Okay. So it takes some time to stop.
It does take a few seconds to stop.
Yeah.
So, and I imagine multiple people are probably experiencing that too because everybody has the default runtime going. So everyone's going to need, if you have a runtime on right now, you're definitely going to want to stop that. We're going to delete the runtime so that we can choose the Bioconductor environment. So let me see if I can see. For those of you who paused your runtime, would you please speak up when it looks like it's stopped running so that I can know when people are kind of ready to go?
Mine stopped.
Yours is stopped. Okay. So if you've, since you already had a runtime created, you're going to need to delete that runtime. To do that, you would go to, you would open up, sorry, let me go back here. Click on your runtime widget in the upper right, and then go to delete runtime at the bottom of your runtime configuration window. And just select delete. Now when you look at your runtime, it should say none on it. And then the next thing we're going to do is click on the widget one more time. And now from the environment drop-down menu, we're going to select the R Bioconductor option under Jupyter Environments. One thing I want to point out about selecting the runtime environment, if you ever wanted to know what's installed on that environment, you could go to what's installed on this environment. This is handy if you want to see what kind of packages are available, particularly Bioconductor packages. When you click on that link, it'll default you to the Python packages in the environment. And if you wanted to know about Bioconductor packages, you could switch over to the R language and see them installed here. If you don't see a package installed on your environment, especially for Bioconductor, that you want to use, you don't need to worry. You can install other packages, and I'll show you where you can find instructions to do that. So our Bioconductor should be selected for your environment. Your compute power should be default, and you can select create. All right. While that's creating, I just want to point out a couple of features here on the dashboard and give you a little overview of what we're doing today. So first of all, I'm going to go into the Notebooks tab just to show you here. This workspace features two Bioconductor packages. One is Single Cell Experiment. This is a commonly used package for manipulating and storing single cell sequencing data. And the other one is EdgeR. This is a commonly used package for differential gene expression. As we're going through these two notebooks today, some of the things you're going to learn about is how to interact with your terminal. We're going to be pulling in some data into your virtual machine. So I'm actually going to show you how to go into the terminal of that virtual machine. In single cell experiment, we're going to learn about single cell experiment objects, and we're going to be plotting out some single cell data into a t-SNE. With EdgeR, we're going to perform differential expression analyses and actually visualize it. Let me go to the dashboard of this while the notebook is starting up. I probably should have started with this, but for those who don't know anything about Bioconductor, Bioconductor is a suite of open source tools that are primarily written in R for high throughput genomic analyses. If you want to learn a little bit more about the notebooks in this particular workspace and some additional Bioconductor packages that this workspace features, you can scroll through the dashboard and look at that under each of the sections here for the different notebooks. I also want to point out that the dashboard contains instructions for what we just did in terms of setting up your runtime configuration and looking at installed packages. And if you, again, don't see the package of interest in here, you can use this Bioc Manager install command in your notebook to install any additional packages. And you'll also find a link here to a README where you can see further instructions about installing packages. The other thing I want to point out on the dashboard of this workspace is the Google bucket. We had some questions earlier about, can you upload publicly available data to your Workspace Google bucket? And the answer is yes. I do want to remind everyone that every time you generate a workspace, you generate a Google bucket storage that goes along with that workspace. So this ID will be unique to my cloned workspace. All right. Let me go ahead and navigate into our Notebooks tab again. And before I continue, let me just check and see, are there any questions up until this point? Hopefully everyone has the notebook runtime creating. And if you don't, let me know. Okay, and mine just finished creating. So hopefully everyone else's is going to finish here. What's that? Oh, I thought I heard a question. Okay. All right, so let's go ahead, and today we're going to start off with the single cell experiment Jupyter Notebook. So let's go ahead and click into it. And again, when you initially open a notebook, as Ali pointed out, you're in preview mode. To actually run code blocks, you're going to need to go into the edit mode of that notebook. And you can only do that when your notebook runtime is actually started up. Okay, once you're in the notebook edit mode, you can view the table of contents to kind of get a sense of what we're going to be doing today with this particular Jupyter notebook. Again, we're going to be looking at some data that is in the form of a single cell experiment object. And a single cell experiment object is just a data structure that allows you to easily manipulate single cell data. So if anyone's actually doing single cell analyses, this is an incredibly useful package because it works with over 70 plus other bioconductor packages for analyses. This particular notebook was designed based on a vignette that you have access to by clicking this link here, if you're looking at my screen, which has more details on using this particular package. And modifications to this vignette were made by Vincent Carey. The data that we're going to use for this particular notebook is already stored in a bioconductor package called RNA seq. It's Allen brain data, and specifically it consists of gene level counts from 379 mouse visual cortex cells. The data for these cells is already provided as a single cell experiment object, and today I'm going to show you kind of how to view that object and manipulate it a little bit more. All right, so before we start running code blocks, you'll notice if you scroll down that this notebook already has the outputs from when it was made and showcased. So I want us to run these code blocks together in real time. So we're gonna clear all this output and start fresh. And to do that, we're gonna go up to kernel at the top of your Jupyter notebook, and you're gonna hit restart and clear output. And then you'll be prompted and you're going to select restart and clear all outputs. All right, so that way we're all kind of on the same page as we're going through this. All right, so the first thing we're gonna do is load some necessary packages to use this notebook. Again, we're gonna be loading the single cell experiment R package, but also the single cell RNA seq and RT-SNE package. So go ahead and run those. And when you do, you won't expect to see any output. And then the next thing we're going to do is examine the Allen brain data that I described, those 379 visual cortex cells. And we're going to rename this as SE, single cell experiment, and take a look at what this looks like. This is data that's already in the form of a single cell experiment object, so it's already in that data structure. So go ahead and run that code block. I've been using the run button here to run cells. I should mention that you can also use control return if you're working on a Mac as a shortcut. Okay. So hopefully everyone is here. So the output of this first code block just tells you a little bit about this single cell experiment object. You can see it says class single cell experiment. The dimensions of this are 20,816 by 379. That's because there are 20,816 genes listed in this object and 379 mouse visual cortex cells. The other thing I want to draw your attention to is the assays here. You can see that there's 4 types of count data represented in this object. There's top hat counts, cufflinks, FPKMs, RSEM counts, and RSEM TPMs. The row names you can see are our genes. Here are the gene IDs. And the column names are going to be the cell IDs for each one. You'll also notice this alt experiment names. This is a place where you can store additional metadata that's related to single cell analyses. In this case, it's showing you ERCC. That's a form of RNA Spiken. And you could also store CRISPR tags or antibody tag metadata in this alt experiment names here.
In my data, I have only the top hat counts.
So that's the next cell down. So you might have jumped ahead of one cell. Let me go back up, I'll show you. So this cell 2 only has reprocessed Allen data. And the next step then is we're going to essentially write a new single cell experiment object that only looks at top hat counts down here. So that's the next step. So it's okay that you ran that first, but in general, we try to run notebooks in order. All right, so let's go ahead and do that. So again, today we're going to be only interested in looking at the top hat count data. So I can write a new single cell experiment object by specifying the top hat counts assay here. And now when we look at the output, you'll see the difference from the first output that under assays, you no longer see the other count info, such as the FPKMs or the RSIM TPMs. Everything else should look very similar to the first output we saw. But that just gives you a sense of how you can rewrite the single cell experiment object to be more specific if you wanted to. Now this next section shows you how to manipulate different Spiken data. Feel free to go ahead and run these cells. I'm actually just going to, I think I'm going to skip over this one for today and go straight over to accounting for sequencing depth, just in the interest of time. But again, feel free to play with this if you're interested in seeing how it's presented. It's just some example code to show you how to manipulate that data. So now I'm going to section 1.5, accounting for sequencing depth. Often we want to account for the total gene reads that a sample has and normalize for that. And to do that, we would take the sum of the total counts for each individual cell sample. So here you can see some code that allows you to specify those column sums of our top hat counts and call it under this field size factors. So let me show you what this looks like. All right, so here you can see now a sample of the cell IDs and the total cell counts. So if you want it to normalize gene expression, this is just a way you could do that working with a single cell object. The other thing I wanted to show, I'm gonna do this here, is if I look at my single cell experiment object again, I just typed in SCE in this code block. You see, if you go down to column data names, that size factor has been written to our data structure. All right, and again, here's some code that you could do the same for your Spiken, mRNA Spikens, which I'm just going to skip over for now. Okay. The next thing that this notebook shows us how to do in section 1.6 is to create a subset of our data. And specifically what we want to do is log transform the data to standardize for differences in gene variation. And we're going to take the top 100 most highly expressed genes and order them from in descending order. We're preparing that data so that we can then plot it in a t-SNE plot. So in section 1.6, go ahead and run the code block there to create this subsample. All right, and when you do that and look at your new single cell experiment object, you should now see that the dimensions are 100 because we've taken the top 100 genes. And again, we're still looking at our 379 cells. Now that we have a subset, we're going to use a different package, this RT-SNE package, to do some data reduction. Specifically, we're going to do an initial Pca analysis, and then we're going to prepare our t-SNE data. And then we're going to put both the Pca and t-SNE data into our single cell experiment object. So let me show you what that looks like. Go ahead and run the code block in 1.7. Okay. So again, this should look pretty familiar to what you've seen before, except now, when you look at your object, you see this reduced dim name section now contains our Pca and t-SNE data in it. Okay, so we can make a plot of this t-SNE data, and we're going to color that plot by the cell type, which is this primary type here. So let's go ahead and run that. I heard that. It's a bad cluster. So you can see this plot is not very pretty, and also there's no key here to tell us what these different cell types are. So we're going to use Ggplot to make a better looking plot, and we're going to create a new data frame with the primary cell type included in it. So go ahead and run the new data frame cell block. Okay. So in this data frame, again, you can see our cell samples here. X1 is going to be our factor 1 on the x-axis of our new plot that we'll do in a second. X2 is the y-axis of our new plot. And the type listed here in the last column is the cell type. All right, so the last two options here will actually just make the new plot using Ggplot. And now we can see a slightly improved visualization of our data with a key on the side. Okay. Now, one thing that hopefully everyone has gotten this far, and please tell me if anyone has questions right now or wants some more time to get through these different fields. Okay. I also wanted to give you a chance to create a new cell block and look at some of your data in here. So I'm going to scroll back up, and I just forgot to do this earlier. If you could go back to section 1.3, the creating a single cell experiment object. Here, this is where we specified the top hat counts. I'm just going to wait for everyone to catch up here. If I wanted to look at what the count data actually looks like, I could do that. So I'm going to go up here to cell, and I'm going, oh, sorry, to insert, and I'm going to say insert a cell below. All right, and then I'm going to specify assay SCE 1 to 5, 1 to 5. So I'm going to take a look at what the count data is for the first 5 cells and the first 5 genes. So if you're following along, go ahead and type that in and then run that block. And here you can actually visualize what your count data looks like within this single cell object. So again, here we have our gene IDs going down the column and our cell IDs going across the top rows. And you'll notice the majority, or a big proportion of the counts are 0, which is expected because most cells won't be expressing every gene. Okay. All right. If you want more information on using a single cell experiment, I do encourage you to go to Bioconductor's page for it. A link to it is at the bottom of this tutorial. And if there aren't any questions, I'll switch gears to our EdgeR notebook to do differential expression analysis. Are there questions? I can't tell. Let's see. Oh, okay. Yep. So I saw one of the questions was how to install packages, and it was answered on the chat. And I do want to mention that that code, I'm gonna, that same code, that Bioconductor Manager install code is also on the dashboard of this workspace. So if you needed just a reminder of it, in addition to going to the Bioconductor documentation, it's there too as well. All right. Any other questions? Okay. Then I'm gonna go now. I'm gonna show you all a shortcut for getting back to your notebooks. If you go up to the top of your workspace here, you see that you can actually select the notebooks words at the top there. That'll take you back to your notebooks tab. And I'm gonna open up the EdgeR notebook now. Okay, let's see if I can exit out here. Okay, now, because both of these notebooks run off of the Bioconductor Runtime Environment, there's no need for me to change my runtime. I can just leave it going right now and click into the edit mode of the EdgeR notebook. Okay, so this notebook was designed to look at differential expression between cells that lack BOC1, a transcription factor involved in oxidative stress pathways, and control cells. These are human cells, and they were taken from a publicly available geodataset. So someone was asking earlier about uploading geodata. While this isn't necessarily maybe what you had in mind, maybe you're thinking of FASTQ files. I took this counts data from Geo and I put it into a public Google bucket. So I'm gonna show you a little bit today about storing things in Google buckets versus your virtual machine. So before we get too far into that, I do want to go ahead and just load some packages here for EdgeR. So go ahead and run the first code block. And the only output you should get is this loading required package Lima. Okay. Now what we need to do is I mentioned that we're using publicly available counts data. Currently, that data actually lives in a public Google bucket. I took that data from Geo and I put it into a Google Cloud Storage bucket. Hopefully you can see my screen here. It's Terra Featured Workspaces Bioconductor. And it's just a text file called counts.txt. So I'm showing you right here the Google bucket view. And if I were to click on it, here you can see what it looks like. It's got ensemble gene IDs, and then we have 3 control samples with the C here, SGC1, C2, C3, and 3 BOC1 knockout cells, this SG21, 22, 23. So what we need to do is call this data into our notebook, which is running on a virtual machine. So I wanted to take this opportunity to kind of show you what your terminal looks like for your virtual machine. So to access the terminal, you go up here to your runtime widget and go to the left and you'll see this open terminal. This is going to open up your terminal on a new tab. And here you can basically treat this like you would treat terminal on your local machine. You can print your working directory. You can see what's in it. I'm going to change into the notebooks tab of my terminal. And here you can see that I have a folder dedicated to my workspace, Bioconductor July 09 LK. So I'm going to change into that. And then you see that there's 2 modes available for that workspace, edit and save. And I'm going to change into the edit mode. Okay, now when I look in my terminal into this directory, you see my 2 notebooks that are available on the workspace. But what you don't see here is a counts.txt file, which is what the Jupyter Notebook is going to need to access to actually run the differential expression analyses. So if I go back over to my EdgeR tab, I'm going to run this second code block here. We're in section 2.2 now of the EdgeR notebook. I'm going to run this code, which is going to copy, using the gsutil command, that counts.txt from the public Google bucket into my virtual machine. So I'm going to run that. And I'm going to run the next file right here, the list.files as a sanity check. So here you see on the output of my Jupyter Notebook, I'm listing the files that are currently on this virtual machine. And if I went back over into my virtual machine and did ls, you again would see it here on the virtual machine. So hopefully that gives you a sense of being able to pull things in and explore them using your virtual machine a little bit, or using the terminal, I should say, of your virtual machine. The other thing I wanted to mention about bringing in data is I chose to put this counts data into a public Google bucket, but I could have easily uploaded it to my Workspace Google bucket, which is what people have been asking about. So let me just show you where you would do that. You would go to your Data tab, go to Files, and then just choose the Upload icon down here. And I could just directly upload that same text file to my Workspace Google bucket storage. I'm going to go back to the EdgeR notebook now, just to show you there is some sample code here for how to import then, instead of importing from this public Google bucket, how to import from your Workspace Google bucket. It's the same command, only you just need to use your Workspace ID, which is found on the dashboard of your Terra workspace. I can show that really fast too. If we go back to the dashboard of the Terra workspace, of your cloned workspace, you should see the Google bucket ID right here at the bottom and the option to copy it. Okay. All right, are there any questions up to this point? Okay.
How would you create a Jupyter notebook if it was like something different? Just
how to create a Jupyter notebook? You could just do that from the Notebooks tab. Here you could say create a new notebook, and that's how you would create.
Oh, okay, okay, thank you.
Yeah, that's a great question. Okay. All right, so now I'm going to go through the remainder of the EdgeR example. So the first thing we're going to do is just read that counts.txt file as like a sanity check. Just to make sure that it looks right, that we see our ensemble IDs and our different samples here. The next step then is going to be to prepare a data frame. So I'm going to run this code. And this is an opportunity where we can actually rename our samples if we want to, and rename the column headers here if we wanted to. So this is just some code to let you do that. All right. In section 2.5, there's also some code here that allows you to make sample subsets. So let's say you have a counts file that had more treatment groups in than you necessarily want to analyze. You could create a subset of your data using this example code here. So we're just going to run that. And in this code block, we also specify our treatment conditions as group. So again, I only had 2 conditions. I have control C and treatment T. All right, and then we can create a new data frame with that subset. Now to use EdgeR, you have to create a DGE list object. That's the format that your data has to be in. So we're going to do that with this next code block, as well as filter the data so that lowly expressed genes are removed. We're going to normalize the data using the TMM method and take a look at the data with box plots. If you have any questions about this process or what these steps are doing, I am going to point you to the EdgeR user manual, which has fantastic description of each of these steps and the EdgeR functions. So go ahead and run this code, and hopefully you will see a box plot after running it, showing each of your different samples. The next step then is we're going to create a model matrix where we're going to assign the treatment groups to each of our samples. So go ahead and run that block. And what you will get in return is basically a matrix where you have your samples and your 2 treatment conditions. A 1 indicates that a sample is assigned to a particular condition, a 0 indicates that the sample is not. Okay, now that you have your model matrix prepared, you can actually do some dispersion, estimate dispersion, and test for differential gene expression. So in EdgeR, we do 3 types of dispersion. We estimate common, trended, and tag-wise dispersion. So go ahead and run these blocks. This one as well. And you should see a graph of your dispersion here, and you can see if your trended matches your tag Ys. Okay, the next thing we're going to do is fit a negative binomial general linear model for each tag. And we're also going to assign our contrast. So specifically, we want to compare our treatment to our control. So I'm pasted in here, T minus C. So we're just telling it what we would like the comparison to be. And then we're going to use a likelihood ratio test to look at differential expression. So go ahead and run that. And you can also look at, run the next block, which will actually return the differentially expressed genes. Okay. So in the output of this block, you should see that 1,933 genes were downregulated in our treatment, 9,620 genes were unchanged, and 1,794 genes were upregulated with our treatment, so with our BOC1 knockout. You could also change the p-value and adjust your method. Here it's set for false discovery rate, and rerun it. This was already the default setting for us, so it doesn't change anything. And then you can look at your top tags, so that the top genes that were differentially expressed. And again, it returns the ensemble IDs here, along with our FDR for those genes. Okay. And lastly, if you wanted to plot the differential expression, you could use this plot MD function. This is going to create a smear plot. And you can see that the upregulated genes are in red and the downregulated genes are in blue. Okay. So again, I'm going to show you now where the Bioconductor Manual lives. If you follow this link here, it'll redirect you to the Bioconductor page and for EDAM. And if you scroll down, you can see an excellent user guide PDF. And this will walk you through the steps that we just did. So if you're interested in doing differential gene expression with EDAM, this is a fantastic resource to use. And again, if you are interested in using this notebook for your own counts file, you can use the steps that I showed you today to go ahead and upload it to your Workspace Google bucket and get started right away. All right, are there any questions about using Bioconductor in Terra?
So listen, how will you extract this counts, and once we get the result, how will you extract and save this gene list?
Yeah, so you can use GSUtils. There is code that you can do to actually export. If you're familiar with coding in R, you would do what you would normally do to create a text file, export a list to a text file. I actually don't know off the top of my head how to do it. I'd have to look it up right now. But once you've created created a text file with this output, again, you can use that gsutil command to move it from your virtual machine to, say, your Workspace Google bucket. And I could show you an example of that if you would like. Let me go back and, oh, sorry, go ahead.
No, please go ahead. Please go
ahead. Okay. So if, again, I don't know how to export this list using R commands off the top of my head. So if anyone does, please speak up, because I can just do it then for you guys right now.
Write.table.
What is that?
Write.table, and then the name of the file.
Let's see. Well, do I have to, let's see. What do I do after, like if I wanted to export, for instance, this list of top tags, would I just do write.table top tags?
And yes, with the file name at the end, comma, at the end say comma, after LRT close bracket, comma.
Top text?
You need to write a file name equal and then put the name in the
Right here?
Quotes.
Right
Maybe
here?
file, I don't remember, and quotes.
File, only file.
File and quotes. Does that look right for those who are looking?
And
Missing closed brackets.
In closed brackets. What, the file needs to be in closed brackets?
No, the function name.
you have the brackets in the closed before file,
Yes.
but you have to remove it.
Exact. Someone
Yeah.
posted the exact.
There you go. Perfect.
Yeah. There you go.
Perfect. Okay. So let me run this really fast for you and hopefully. Okay. So in theory, this worked. It didn't give me an error, so that's good. So now if I go back into my terminal here and type ls, haha, there it is, top.txt. So now, now if I wanted to move that to my Google bucket, I could using gsutil. So let's do gsutil cp. I need my Google bucket ID, which I can find on my dashboard. Copy that bucket name and go back in here and paste it. And I'm going to move, oops, I need to do cp. Sorry, we're gonna copy the top.txt. All right, that should do it. Okay, so now if I go into the data tab of my notebook and go to files, there it is, top.txt. So now this is saved in my Workspace Google bucket. And I could just download it if I wanted to. And here it is. Download. There it is. So, so hopefully that gives you a sense of doing that.
Yes, thanks.
Yeah, great.
Write table has a parameter so that you will not put quotes on each value, which is a bit annoying.
No.
So if you write help write table, then it says what's the parameter.
Okay. All right. Are there any other questions? Well, thank you all very much for tuning into that and for helping with the R code as a group. That was fun.
How is all this connected to the Galaxy?
So the Galaxy wasn't quite ready to go yet for this presentation. So this actually, what I've shown you today is not related to Galaxy. I know that was on the original agenda, but that's still coming in the works.
Okay,
Who is maintaining this platform?
The notebook or the workspace? The workspace is this.
Terra.
It's on Terra, and yeah, and it's maintained by the Broad.
Because I've noticed that there is an option of download as, which is useful if we want later to see what we did today. And downloading as R or HTML works fine, but when you download as notebook, it opens two download windows in parallel.
Say that again. When you download the window, it does what?
So there is under file, there is an option download as.
Oh, okay.
And when you choose download as notebook, which is the first option, it wants to download it twice.
Oh, okay.
And this does not exist with the option for an R note or an HTML.
Okay. I would need some assistance in changing that, or I may ask Jason or Tiffany to jump on if there's something we can do about that.
Yeah, I'll take a look,
Absolutely.
see what's going on.
Thank you.
Is there a way that users can report problems to a support site?
Yes, absolutely. So if you find problems, let me share my screen again here really fast. Share screen. Okay. If you go here and you go to support, there is a community forum. There's also the request a feature that I just skipped right over. But so on this community forum, you can post questions. There's also featured workspace specific feedback that you can give here. And additionally, if I go back in here, if there's a particular feature that you would like to request, there's also this request feature section here.
Yeah, there's a link to the community forum page that you just showed, Liz, on actually in the wrap-up slides, which I'll be covering in just a few minutes. So folks will have access to that quite easily.
Great. Oh. Okay. Well, in that case, I will turn it over and thank you all for watching.